Good afternoon. It is an honor for me to share with you the results of our work entitled Dosimetric and Biological Characterization of a Three-Dimensional Culture Model for the Evaluation of Bystander Effects Induced by Radiopharmaceuticals. This work is the result of a collaboration between the Laboratory of Development and Research of Radiopharmaceuticals of the National Institute of Nuclear Research, the Autonomous University of Mexico State, the Autonomous University of Mexico, and the Ministry of Science, Humanities, and Technology. The use of targeted radionuclide therapy, TRT, has grown significantly in recent years due to its remarkable therapeutic efficacy and versatility. Although the radiopharmaceuticals using TRT are highly specific, they also produce effects in cells that were not directly affected by the radiation. Following radiation exposure, different signaling pathways are activated, promoting the release of soluble factors that remodel the tumor microenvironment and modify the biology of neighboring and distant cells. Given the complexity of the tumor microenvironment, culture models have been developed for in vitro study that seek to mimic the structural complexity of tumors. Thus, three-dimensional cultures are an indispensable tool for preclinical research on radiopharmaceuticals, which have revolutionized translational cancer research. Three-dimensional cultures allow the distribution of radiopharmaceuticals to be predicted in environments that mimic the pathophysiology of real tissues, facilitating validated dosimetric estimation. The research and development of new radiopharmaceuticals requires the preparation of the standardized three-dimensional culture models that enable advances in personalized medicine through the study of radiobiological responses in controlled environments that consider the presence of metabolic gradients, cell-cell interactions, and tumor heterogeneity. The use of three-dimensional culture models can be leveraged to understand the mechanisms of radioresistance and their relationship with radio-induced gene expression. Given the wide applicability of three-dimensional culture models and the growing interest in developing new and improved radiopharmaceuticals with appropriately characterized molecular targets, we set out to standardize a method for preparing a three-dimensional culture model that accurately represents the pathophysiological conditions of tumors. This dosimetrically and biologically well-characterized model will enable the evaluation of the bystander effects triggered by the radiopharmaceuticals developed and produced at the National Institute of Nuclear Research as part of their preclinical characterization prior to translation. Sodium alginate was used to prepare the culture model because it is an inexpensive and biocompatible scaffold. The optimal conditions for preparing the model were established through factorial analysis. It was established that the ideal conditions for preparing a three-dimensional culture model that is resistant, biocompatible, and suitable for analyzing the radiobiological responses induced by radiopharmaceuticals targeting the prostate-specific membrane antigen, such as lutetium-177 high-PESMA, were as follows. A 4% sodium alginate solution mixed in a 1:1 ratio with LNCAP and PC3 prostate cancer cells in suspension. This mixture was polymerized with a 4% calcium citrate solution at room temperature. As shown in the figure, batches of the alginate beads were prepared and evaluated for shape, size, sphericity, mechanical stability, and encapsulation efficiency. The viability of the cells inside the beads was also evaluated over a period of 96 hours using various techniques, confirming that the cells maintain a viability rate of over 90% without losing their ability to proliferate. In summary, the average diameter of the beads was 3.71 millimeters, so that the cells embedded therein are located at a distance of 65 micrometers from each other, maintaining a viability percentage greater than 97%, with a doubling time of 36 hours. To continue with the dosimetric characterization, the percentage of radiopharmaceutical uptake and internalization was experimentally determined to be 7% for LNCAP target cells and 0.99% for PC3 cells. Given that in three-dimensional cultures the cells do not stretch on the substrate and remain round, it was necessary to determine the cell size under three-dimensional culture conditions. Using fluorescence microscopy techniques, it was estimated that the average diameter of the cell body is around 10 micrometers and that of the nucleus is 8 micrometers. Using these experimental values, the absorbed radiation dose was estimated using the TOPAS-nBio code. To simulate particle transport, a spherical geometry was generated containing spheres that simulated randomly distributed target and non-target cells. The fuel emission spectrum of lutetium-177 was considered for the dose calculation. As shown in this representative image, high, medium, and low energy particles were considered, with one cell as the target and the same cell, neighboring cells, and the medium as sources. As shown in this table, the average absorbed radiation dose received at 96 hours by the nucleus of the LNCAP target cells was 1.5 gray, while the PC3 cells received an average dose of only 0.06 grays during the same exposure time. In other words, the dose deposited was 24 times higher for the target cells compared to the dose received by the non-target cells. To evaluate the cytotoxic effect produced by these radiation doses in the three-dimensional culture model, a methodology was established based on differential staining of cells with fluorescent markers, such as green cell tracker, to distinguish between cell types, and propidium iodide to recognize dead cells. These yielded kinetic images of the radiopharmaceuticals' cytotoxic effect on both cell types, and made it possible to estimate the percentage of cell viability over time. By correlating the measured biological effect with the dose deposited in each cell type, the following survival curves and their corresponding radiobiological parameters were obtained. The survival curves show that in a simulated prostate cancer tumor environment, where prostate cancer cells with heterogeneous levels of PSMA marker expression converge, the radiopharmaceutical will have a considerable and specific cytotoxic effect on target cells, but will also alter the viability of non-target cells to a lesser extent. However, the decrease in PC3 cell survival observed does not appear to be the result of the absorbed radiation dose, which was only 0.06 grays, allowing us to assume that the effects observed are promoted as a result of the bystander effect. Although these results are preliminary and must be confirmed and evaluated in a more complex coculture, they demonstrate the usefulness of the standardized three-dimensional coculture model for evaluating bystander effects. This opens up the possibility of continuing to study the mechanisms through which the cytotoxic effect on non-target cells occurs. Therefore, the results shown allow us to conclude that the model prepared is suitable for radiobiological evaluations of targeted radiopharmaceuticals under simulated tumor conditions. These evaluations take into account cell heterogeneity and communication, as well as non-targeted and distant effects. Thank you for your attention. If you have any question, I will be happy to answer them.